Hello, the topic today is cell biology, and we will be going to solve the question from GATE 2020 Excel Microbiology section, question number 9, where we have to match the cellular organelle in group 1 with its function in group 2. So in group 1, there are 4 organelles: Golgi apparatus, nucleolus, peroxisome, and proteasome. And group 2, there are the functions which are lipid degradation, protein sorting, and ribosomal RNA synthesis. So in order to answer this question, we have to know the features of each of these organelles, which we will see one by one. So first we have the Golgi complex. So the functions are: secretion is the major function of Golgi apparatus. Then we have modification and packaging of various materials in the vesicles. Now this is the Golgi apparatus, which is somewhere closer to the endoplasmic reticulum. This is the endoplasmic reticulum, and this green one is the Golgi apparatus. The Golgi apparatus consists of tubule-like structures and also the vesicles. And these vesicles contain substances which are needed to be secreted. So, what kind of substances? We have proteins which are secreted, then secretion and synthesis of glycoproteins. Glycoproteins are proteins conjugated with carbohydrates. Then, Golgi complex also helps in the formation of cell plate, cell wall, and new plasma lamina during the cell division. So, cell wall, cell plate, it happens in the plant cells. Formation of primary lysosome and sperm acrosome. So primary lysosomes are acidic compartments which are formed, or they arise from the Golgi apparatus, and then they get modified. And sperm acrosome. So sperm contains acrosome, which contains various enzymes which are needed to degrade the extracellular matrix leading to the ovum. Participates in recycling of membranes. So the older lipid bilayer is removed, and the Golgi apparatus synthesizes new lipid bilayer, which by using the secretory vesicle they get fused to the plasma membrane. Facilitates glycosylation. What is glycosylation? It is addition of carbohydrates to the protein. Liposylation, formation of lipoprotein, or uh, a formation of proteins conjugated with lipids. Sulfation, addition of sulfates, and phosphorylation, addition of phosphates. So various kinds of protein modification happens in the Golgi apparatus, as well as they can be secreted. Next, we come to nucleolus, where the major function is ribosomal RNA synthesis, or rRNA synthesis. We have three kinds of RNA. We have mRNA, which is the messenger RNA, which codes for the proteins. Then we have tRNA, or transfer RNA, and then we have rRNA, or ribosomal RNA, which constitutes the RNA which is present in the ribosomes. So the function of nucleolus, it takes up about 25% of the nuclear volume. Here you can see the blue region is the nucleus and the green portion is the nucleolus. And it performs extensive synthesis of rRNA, which is the component of ribosome, and it is composed of RNA, DNA, and proteins. Next, we come to peroxisome. So, peroxisome is an organelle which is present in both plants and animals. The function in animals or humans is mainly the beta oxidation or degradation of fatty acid. So, this is one of the major function. Then we have then the peroxisome also contains catalase. So, what does catalase do? It is an enzyme that can reduce hydrogen peroxide and other oxidative radicals. So it is a free radical scavenger, and it performs or reduces hydrogen peroxide. Then it also helps in the synthesis of cholesterol. Enzyme involved is HMG-CoA. Synthesis of bile in the liver, synthesis of lipids to produce myelin, involved in detoxification of alcohol from the liver and breakdown of excess fluid. So these are some of the functions of peroxisome in animals, and in plants they have some special functions. That is, they perform in the glyoxylate cycle, and they also perform the photorespiration in plants. So photorespiration is the respiration which happens in combination with photosynthesis, and in plants also it helps in the degradation of purine. So glyoxylate cycle and photorespiration are the special functions in plants. This is the structure of the peroxisome. Lastly, the organelle mentioned was proteasome. So proteasome is the site for protein degradation. So this is the normal structure of the proteasome. This is the normal structure of proteasome. It contains a barrel kind of structure which is called a 20s core particle, and it contains caps that are called the 19s caps. So these are the caps. There is a barrel and there are two caps at two ends. So 19S regulatory subunit is used for protein recognition targeted to degradation. So whatever proteins are tagged for degradation is recognized by the 19S subunit, and it has ATPase domain which helps it to unfold the substrates and translocates them into the 20S particle. So this is the 20S core particle. The 19S subunit recognizes the proteins to be degraded, it unfolds them, and then it transfers it to the core 20S subunit. Now, the energy necessary to extract ERAD substrates out of the AR membrane. So, ERAD is endoplasmic reticulum associated degradation. So the substrates which are tagged in the endoplasmic reticulum, they have or some misfolded proteins which are mainly tagged in the endoplasmic reticulum. They have to be extracted out of the ER membrane, and 19S subunit, which with its ATPase activity, is required to do that. We also have the 20S core particle in the proteasome, and it is the cavity in which the targeted proteins are degraded. Alpha subunits are dark and beta subunits are light. So this is the major function is protein degradation. And how are the protein recognized by for degradation? The proteins are tagged. So say suppose this is a protein, so they will be tagged by certain molecules that are called ubiquitin molecules, or the proteins are ubiquitinated. So, if there is a long chain of ubiquitination that is recognized, that acts as a marker for degradation, and that is recognized by the 19S, which then translocates it to the 20S core subunit. So after having this much information, we can now answer this question, that is Golgi apparatus, nucleolus, peroxisome and proteasome, and group 2 lipid degradation, protein sorting and ribosomal RNA synthesis. So Golgi apparatus is concerned with protein sorting. So all kinds of modification, secretion and sorting happens in the Golgi apparatus. Nucleolus, as we read now, that it is involved in the ribosomal RNA synthesis or rRNA synthesis. Peroxisome was involved in the degradation of fatty acids. So fatty acid is a lipid, and therefore it is involved in lipid degradation. And lastly, proteasome. I said that proteasome recognizes proteins that are marked for degradation. So protein degradation happens in the proteasome. So the correct option will be P is 3. So we have A, B and D. Then we have nucleolus, which correct option is 4. So option number B, and R is 1 lipid degradation, S is 2 protein degradation. So the correct option is option number B. Thank you.